Shortly after fertilization, the fertilized egg or zygote begins dividing. By about 4 days after fertilization, a solid ball of cells called a morula forms. The cells continue to multiply, and by day 5, the cells separate into two groups, the inner cell mass, which will become the embryo, and an encompassing sac called the trophoblast, which will develop into the placenta. Trophoblast cells secrete fluid, creating a cavity, the blastocele, with the inner cell mass at one end. At this stage, the mammalian embryo is called a blastocyst. The cells of the inner cell mass eventually form two layers, the epiblast and the hypoblast. The epiblast is thought to contain the cells that will generate the actual embryo. Critical to the early development of all vertebrate embryos is the process of gastrulation, which occurs between days 13 and 19. Gastrulation results from the movement of cells toward the midline and then forward along the midline, forming a groove called the primitive streak. The end result is the production of the three primitive cell layers, the endoderm, the mesoderm, and the ectoderm. The nervous system develops from the outer layer, called the ectoderm. As this cell layer thickens, it becomes a distinct epithelium called the neural plate. Uneven rates of cell division form a groove called the neural groove that will become the midline of the embryo. Ridges of ectoderm continue to bulge on both sides of the midline. The tops of the neural ridges eventually come together, forming the neural tube. The interior of the neural tube becomes the fluid-filled cerebral ventricles of the brain, the central canal of the spinal cord, and the passages that connect them.